I'm Scott Podolsky, and I help to co-organize this departmental seminar series. And it's my pleasure today to get to introduce my friend and colleague, Carol Mitnick. As I'm sure many of you know, Carol has long played a leading role in bringing concerns with social justice and health equity to global health, and especially to the treatment of tuberculosis. Carol received her BA in political science and French from the University of Rochester before receiving her master's, and by 2001, her Doctor of Science degree from the Harvard School of Public Health in international health epidemiology and ecology. She'd already started working with Partners in Health in 1996 and joined our department in 2001, where she's now professor of global health and social medicine. Carol has led in the effort to bring treatments for TB to low and low middle income countries, as exemplified by her key role alongside her departmental and PIH colleagues in several iconic and really paradigm shifting trials in New England Journal of Medicine articles on the treatment of drug-resistant and extensively drug-resistant TB. At the same time, she has long led in the effort more generally to apply clinical trials to the evaluation of TB interventions and to shape global TB policy in accordance with such findings. At the intersection of all of this, Carol is the co-PI of the EndTB and EndTB-Q trials and PIH Director of Research for the EndTB project. As we'll hear from her today, the EndTB observational study, working across 17 countries and examining the experiences of nearly 3,000 patients with the first new anti-TB drugs in half a century, has already informed WHO guidelines for the treatment of MDR-TB. We'll get to hear from Carol on this, and then we'll have time for Q&A, and you can use the Q&A function to ask questions for her. And for those who know others that want to watch this or other videos from our series, you can plug into the HMS Global Health and Social Medicine YouTube channel, which apparently is experiencing exponential growth in usage. So with that, I'll hand this over to Carol.
Great, thanks so much, Scott. I've got to check to make sure I'm subscribed to our YouTube channel. I know I've checked it, but I don't know about subscription. So I'm going to share my slides here. And see if this works according to plan. All right. So thanks so much, Scott. Thank you, Amy, for the help organizing the webinar today and for the opportunity to give an overview on the EndTB observational study.
I don't see your slides yet.
Oh, you don't? That is weird. Okay. Let me try this again, sorry.
There we go.
Okay, hang on. What is going on here? So you see my slides, but it's not in this projection mode. Now
we're good.
Okay. And you see your view, not notes and things, is that correct? Okay. So thanks.
It says slide 1 of 98, but I don't see any notes. I think we're good.
Okay. So I am going to start with some acknowledgements. I cannot emphasize enough how the work that I'm going to share today is literally due to the participation and contribution of thousands, and first and foremost, all of the participants in the EndTB observational study about which I'll be speaking, the teams that worked on this project in all of the 17 countries where the project operated, the National TB programs and other collaborators in those settings. The funding for the EndTB project comes largely from UnitAid, and Médecins Sans Frontières and Partners in Health have also made contributions to the larger project. And we work in partnership with Interactive Research and Development, HMS, of course, Epicentre, the research arm of Médecins Sans Frontières, and the Institute of Tropical Medicine in Antwerp. These are just a few photos from some of our collaborating sites around the world. So today I'm going to give you just an overview, an update on what we did in the EndTB observational study, really what was the problem that prompted this study, what did we do in the study, talk about some of our very kind of narrow scientific findings, and then zoom out a bit to the bigger picture contributions of the EndTB observational study.
I'm so sorry.
No worries, Scott.
No, I'm getting some Q&A that folks are seeing the presenter mode. For some reason I can't see them.
Okay.
Yeah, so they can see. Sorry.
How about now?
That looks different. Soon good. If anyone else can see it, please let me. Since he knows, please let me know. But otherwise, we're good. Sorry, Carol.
Okay. No problem. No problem. I apologize. I thought I had it settled before. I'm sorry. I just need to move so I can see now. Okay. And then I'll conclude by sharing some of the challenges that we still face despite this big effort. So what problems was the EndTB observational study and the larger EndTB project trying to address? So as most of you know and have heard me and others say before, there is a substantial burden of multi-drug resistant tuberculosis, that is tuberculosis caused by strains of Mycobacterium tuberculosis that are resistant to the most important first-line drugs. Of the more than 10 million cases, new cases of tuberculosis each year globally, about 500,000 are estimated to be caused by these organisms resistant to the most important first-line drugs. But only a small portion of those cases actually get tested for drug susceptibility. An even smaller proportion, just under 40%, actually get notified, kind of a step in the process toward being treated. And then of those notified, only about a third actually get treated. And this leads to, at the center of the circle, just this tiny proportion, about 8% of the 500,000 new cases that actually get treated or were getting treated successfully each year at the time that the EndTB project was conceived and started in 2015. The care is really, or was really challenging. It was an 18 to 24 month regimen. Obviously, that is long. It was difficult to tolerate. Specifically, the use of a daily injectable made it really hard to tolerate, and it was quite toxic and only moderately effective in most places. Just to give you a sense of what I mean by only moderately effective, you can see from the small selected subset of studies that treatment outcomes, that favorable outcomes hovered around the 50% mark. When meta-analyses were done, probably with a publication bias, the results appeared to be a little bit better. But I think for me, particularly strikingly in the context of a randomized controlled clinical trial, a pivotal trial that led to regulatory approval of one of the newer drugs I'm going to talk about today, only about a third of the patients in the control arm, so when the treatment is supposed to be at its best, had successful outcomes. And then the toxicity, although it hasn't really been consistently measured using the same endpoints over time, there's a lot of at least anecdotal and some observational evidence that a lot of patients required drugs to be discontinued temporarily or permanently during their treatment. So the standard of care in 2015 was really challenging. There was some hope in the form of two new anti-TB drugs, the first new drugs to come to market since in about 50 years. In 2012, bedaquiline was conditionally approved through an accelerated mechanism by the FDA, and then a year later by the EMA. And then in 2014, delamanid was approved by the EMA, the European Medicines Agency, both for indications of difficult to treat drug-resistant TB. The use of both drugs was cautiously endorsed by the World Health Organization shortly after their regulatory authorizations. What happened was very few people actually got these drugs, even though World Health Organization endorsement is kind of the beginning to adoption of innovation in TB treatment and diagnosis. So the DRTB Stat Advocacy Group estimated that there were about 500,000 MDR-TB patients who could have benefited from bedaquiline and or delaminid between 2015 and 2017, and only about 2% or 11,000 of those 500,000 actually got those drugs. So we had a real problem. We had these great potential improvements for care and a real problem in access. The constraints were many, and they included the cost of the drugs. They're Janssen or Johnson & Johnson, the parent company of Janssen that developed bedaquiline, created a tiered pricing structure so that it was $900 in low-income, high-burden countries for a course of bedaquiline, $3,000 in middle-income countries for a course of bedaquiline, and $30,000 for a 6-month course of bedaquiline in high-income countries. And I did fail to mention, sorry, that both of these drugs were authorized for 6 months of use. And that will come into play later. There was also real fear of generating resistance, and this is kind of emblematic of the TB field where we've had little innovation, very few new drugs. The tendency is to kind of protect the drugs and protect them by not allowing them to be widely used, rather than improving the conditions under which they're used and improving the stewardship of those drugs. There was a very, very high level of fear of their toxicity. And some of that was because there was a lot of unknown. They had been administered to relatively few people by the time they received their regulatory authorization. And in the pivotal phase 2 trial of bedaquiline, there was excess death in the bedaquiline group long after treatment was complete. And so bedaquiline ended up with a black box warning in the FDA and EMA labels. Both drugs do prolong the QT interval. And because of that and this uncertainty, their use together was discouraged by the World Health Organization initially. And I think a really important point that made it hard for people to uptake these drugs in routine care, in addition to all the ones I've already mentioned, is that the populations included in these trials are often kind of cherry-picked. They are not representative of patients with multi-drug resistant TB, meaning there weren't many patients with important comorbidities except for HIV. So not much diabetes, not much viral hepatitis, little substance use disorders. And yet these conditions are common among people with drug resistant TB. So what did we do in EndTB? So the EndTB project had as its objectives to expand access to bedaquiline, delaminid, and some repurposed drugs that were newly identified to have really important activity against TB. And the initial approach was to use those according to extant guidance about their use. So that was in longer regimens. And then we also had as an objective to identify shorter and better, hopefully all oral, less toxic regimens, and then really to generate and disseminate that evidence and to use it to enhance access. So it's a big project. It started as a 4-year project and then was extended twice to become a 9-year project, funded to the tune of $90 million. And as I mentioned, that's mostly from Unitaid with contributions from MSF and from PIH. And it's really, I think, somewhat unusual in that it's a very large research project on the foundation of non-governmental organizations, service providers, partnering with research institutions, working very, very closely hand in hand with high TB burden country ministries of health, patient organizations, and NGOs in those settings. There were 3 objectives. There are 3 studies within the EndTB project, and I'm going to just talk about the observational study today. But just so you know, there is also the EndTB clinical trial, which enrolled 754 participants in evaluation of all oral shortened regimens for fluoroquinolone susceptible TB, and the EndTBQ clinical trial, which is completing enrollment this week, hopefully with 324 participants for evaluation of quinolone resistant MDR-TB. And you can see in this map, the kind of purplish areas are the 17 countries in which the EndTB observational study enrolled. And the mustard colored dots are the sites for the EndTB clinical trial, and the kind of teal for the EndTBQ clinical trial. So you can see there's tremendous geographic diversity in the EndTB observational study. So we also, as part of the planning for this project, we essentially operationalized the cautious recommendations from the World Health Organization, and we operationalized them in perhaps the most permissive interpretation possible in creating an EndTB clinical guide that provided guidance about the optimal combinations of regimens, the optimal duration. The whole project also really adopted Paul Farmer's idea that we needed to address the space stuff system staff and support. So in this case, that meant using the funding that we had received to purchase medications, to be able to provide appropriate lab monitoring, both clinical monitoring and also microbiological monitoring, obviously clinical staff, logistical support for the project, and accompaniment of individual patients as well as physicians and national TB programs in this project. We developed a custom electronic medical record system to both support the patient management during the EndTB observational study and also to support rigorous data collection. It was built on the Bomni platform so that it could be, it's an open access platform and could be used in the countries where the project was enrolling, but also in other places. All the participants provided informed consent to receive either bedaquiline and or delaminid, which was the standard at the time, given that the drugs were relatively new and also given the caution around their use. And all participants also provided consent to use their data for research. There was although the care was really in routine TB programs, we had a recommended monitoring schedule and supported our partners in providing monitoring on this schedule. You can see it was quite varied from, again, the microbiological monitoring that I mentioned to hematology, biochemistry monitoring, and the ever important electrocardiograms because of this heightened concern about QT prolongation with bedaquiline and delaminid, and I might add also with other of the drugs that had traditionally been used to treat drug-resistant TB. The project also established the first multi-country pharmacovigilance system for really standardized, systematic recording, reporting, and management of adverse events during MDR-TB treatment. I alluded earlier to the kind of combination of anecdotal evidence that we had used before to understand that the regimens were really toxic, but it had never been very systematic. And so we implemented this for the 17 countries, and this system established at the Doctors Without Borders operational center in Geneva. The MSF pharmacovigilance unit has also been used for a few clinical trials and is available, and for another operational research study that observational research study that I'll mention later. So we identified a group of adverse events of special interest that were linked to these new drugs, to bedaquiline and delamanid, as well as to important drugs that had been used historically, like the injected agents, the aminoglycosides and polypeptides, as well as the repurposed drugs linezolid and clofazimine that were being used frequently in the observational study. And the pharmacovigilance unit really supported, it was part of the accompaniment package for clinicians and national TB programs to do the recording and reporting, and then to notify to regulators and manufacturers as necessary. Moving into what we learned from the EndTB observational study on a kind of narrow scientific side, we, as I mentioned, enrolled and had consented for use of their data for research, 2,727 participants across the 17 countries. They were relatively young, but there really were no inclusion criteria other than that people have confirmed rifampin resistant or multi-drug resistant TB. So there was no exclusion by age. There was no exclusion by pregnancy or comorbidities. And we're going to talk a little bit about that later. You can see a large proportion had low BMI, and more than half had resistance or had TB caused by strains of Mycobacterium tuberculosis that were not only resistant to rifampin, but also to fluoroquinolones, which were really the most important drugs in treatment for drug-resistant TB prior to the advent of these newer drugs. And then large portion had been previously treated for MDR-TB, and then you can also see that somewhere between 10 and 20% of patients had each of these important comorbidities. So this was not the cherry-picked population of a pivotal trial that I mentioned before, and that's super important for generalizability and for programs to really know what they would be facing when they use these regimens later. And just a little bit more granularity on the distribution of these comorbidities, you can see that there is some variability in their distribution. So for example, hepatitis C was more common in the Eastern European and Central Asian countries, Armenia, Belarus, Georgia, Kazakhstan, while in Lesotho, 79% of the participants were co-infected with HIV, and the diabetes was also somewhat variable, but with some high prevalence in the South Asian countries. And I failed to mention, this is not all 17 countries. Obviously, these were the countries that had larger enrollment in the EndTB observational study, so where the proportion of comorbidities is a little bit more meaningful. So our first kind of major scientific finding is that bedaquiline and or delamanid containing regimens were effective. And this had been known to some degree from the pivotal clinical trials. The results from the bedaquiline pivotal trial were pretty non-controversial in terms of the huge benefits seen in the bedaquiline arm, but the control arm, as I mentioned earlier, had performed really poorly, so that did make it easy to see a benefit. And so we didn't know what would happen in routine care, but what we saw was very strong initial treatment response. So more than 80% of each of these resistance subgroups had culture conversion at 6 months on regimens containing either bedaquiline or delamanid or both. There was more interest perhaps in the delamanid containing regimens because the pivotal trial for delamanid actually had been equivocal, where there was no observed improvement in effectiveness in the patients who received delamanid in a background regimen compared to placebo in a background regimen, although its safety and tolerability profile was very encouraging. So we were really interested in digging into this delamanid question in the EndTB observational study. And one of the early papers that we published was on the 6-month conversion in the delamanid among patients who received a delaminid-containing regimen. And again, about 80% of those culture converted within a relatively short period of time. And these were not selected patients. In fact, there was a lot, the prevalence of comorbidities was even higher among the patients who received delaminid than among the whole cohort. Then another question that was of great interest, I had mentioned the injectable as the standard in the regimen of the time. And initially, bedaquiline or delaminid was added to a regimen containing an injectable, but over time we began exploring all oral regimens, so where injectable sparing regimens. And an important question was whether they did perform as well as the injectable containing regimen. And we found again that the all oral regimens had an 84% probability of conversion at 6 months, and the adjusted relative risk of conversion compared to an injectable containing regimen was very close to 1. There were several sensitivity analyses done, and they all came out the same way. And again, as we began to have longer follow-up period, we started looking at the success at the end of treatment in the full cohort of patients in the EndTB observational study, and still saw around 80% success at the end of treatment. The other thing that we looked at recently at the end of treatment is this, I sometimes refer to this as our back to the future analysis. Basically, the clinical guide that I referenced earlier, and I mentioned its most permissive interpretation of WHO guidance. It basically was prescient and anticipated future WHO, explicit WHO guidance about recommendations for how to combine drugs in an effective regimen. And so we went back and looked at the regimens that had been composed according to what are now today's guidelines for composition of these longer all-oral regimens for EndTB, and found that the results, again, were consistently in the 80% range. There is a hint of a difference where the patients with disease caused by organisms that were susceptible to fluoroquinolones may have had slightly worse outcomes than those with disease caused by organisms resistant to fluoroquinolones. We have to dig into this a little bit more, but this is likely due to having been more aggressive in the treatment of the fluoroquinolone resistant disease, or the documented fluoroquinolone resistant disease. You can also see when fluoroquinolone resistance was unknown, the results were similar to those in the susceptible group. But overall, the results were very, very encouraging in kind of every way we sliced the cohort, and despite all of these comorbidities and this very high burden of prior treatment. Turning now to safety in the EndTB cohort, I mentioned that we had really tried to standardize this so that the results were easier to interpret and ultimately would be easier to compare to results from other studies in the future. And I'm not going to go through these in any great detail. I just want to highlight that we identified the adverse events of special interest for the study as being those that were caused by the drugs really of interest in these regimens. So the events that we know to be caused by linezolid, and in some cases also other drugs, are peripheral neuropathy, optic neuritis, and myelosuppression. The events that can be caused by bedaquiline and also by some other first-line drugs, hepatotoxicity. I talked about the QT interval prolongation and the fact that it can be caused by bedaquiline, delaminid, fluoroquinolone, and clofazimine, not to mention other drugs that patients might be receiving for their comorbidities. And then the other really important group of events are those caused by the injected aminoglycosides and polypeptides, and really represent the reason that we were enthusiastic to develop injectable sparing regimens. So these electrolyte imbalance, which combined with the QT interval prolongation, can be really worrying and dangerous. Acute renal failure and hearing loss are the adverse events of special interest. So in this slide, this is kind of the simplicity belies the density, I guess, of this slide. So here we try to represent both the percent of patients who had any particular clinically relevant adverse event, and also the incidence of those events per 1,000 person months of exposure to treatment. And this was important because, again, everybody in the cohort got either bedaquiline or delamanid, but not everybody got linezolid or the injected aminoglycoside or polypeptide. And you can see here that the little red dot and the confidence interval is the incidence. So I'm just going to focus on that for a moment. But there were 1.7 cases of QT interval prolongation of greater than grade 3, meaning 501 milliseconds or longer per 1,000 months of treatment exposure. And there were about 20 times as many the incidence of peripheral neuropathy was about 20 times as high, and hearing loss was about 10 times as high. And then the electrolyte imbalance is again about 20 times as high as the prolonged QT interval, which was really the big concern at the start. So this large observational study was really able to demonstrate the safety of these bedaquiline and delamanid containing regimens, and also offer some insight into what might be the really important clinical monitoring to avoid the dose and duration limiting events. Then specifically when bedaquiline and delamanid were combined, as I mentioned, the WHO guidance didn't explicitly prohibit their combination, but sort of discouraged it. And in about 472 participants in the EndTB observational study, the two drugs were combined, and it was for a range of reasons. Generally, it was clinical discretion and thinking that there was some reason that these patients needed to have bedaquiline and delamanid used together. And you can see that a much higher proportion of those who had combined bedaquiline and delamanid had prior treatment exposure, prior treatment for MDR-TB and resistance to fluoroquinolones than in the overall cohort, and also that the prevalence of these important comorbidities was as high or slightly higher than in the whole cohort. And the people who got linezolid and who got bedaquiline and delamanid concomitantly also got these other important repurposed drugs. So despite the fact that they were a sicker group, a group with more prior treatment and more resistance, their outcomes were again in the 80% range, just slightly below 80%. And also the concerns about having a greater than additive effect on QT prolongation if you put the two drugs together did not seem to be realized. We had a very similar proportion with, actually even lower with proportion with QT prolongation than we had in the whole cohort, and all of those events resolved. I will say very importantly that there was one sudden death and one death of unknown cause, and we can't rule out an arrhythmia or QT, an arrhythmia or torsade de pointe, but we don't have evidence that occurred in either of these patients. And then just thinking, so I talked a little bit already about the importance of doing this observational research to have data on representative populations. Among the other groups that we don't get data from in clinical trials generally are pregnant people and children. And they were not excluded from the EndTB observational study, as I mentioned previously about children, and also just want to highlight that there were people who either started treatment pregnant or became pregnant on treatment and were retained in study. It was not a large number of people out of the 2,700, but we think it's a really important contribution to the knowledge about using these drugs. Women are at heightened risk for developing TB in the peripartum period. And the existing treatments for drug-resistant TB are have teratogenic effects. And so to just say that these new drugs couldn't be used because we don't know whether they're safe or not was not really an acceptable alternative when pregnant women show up and need care for drug-resistant TB. And in fact, bedaquiline is known to be safer than many of the other drugs that are already used in pregnancy. So we're working on a little case series of the women who became pregnant, and you can see the characteristics. So there were 37 of these women, quite young. They had a total of 42 pregnancies. While they were involved in the EndTB observational study, 41 of those were either prior to treatment or during treatment. They had very good treatment outcomes. It's a small group, but 94% had favorable treatment outcomes. And they had a median of 85 days of treatment while pregnant. And these are the birth outcomes. We have not compared these birth outcomes to women without receiving bedaquiline or delamanid who had drug-resistant TB in these settings. So I cannot speak to whether these numbers are higher and lower. We know that TB itself is a substantial risk factor for poor pregnancy outcomes. And as I mentioned earlier, the other treatments for drug-resistant TB are known to be teratogenic. So it is not surprising that there would be some poor outcomes. And you can see that nearly 40% of the women did choose to terminate their pregnancies. They are often counseled to during treatment if that is an option for them. And you can also see there was a substantial burden of low birth weight. But a third of the women had low BMI themselves, and we've already discussed the substantial burden of comorbidities in this population. So the kind of last specific scientific finding I want to talk about is this. It's really two findings I've grouped together. There is a lot of interest in the field of TB and treatment of drug-resistant TB and understanding how to optimize treatment regimens for duration, for effectiveness, for safety. The instruments that have been used to try to do this before are kind of blunt. So in the EndTB observational study, we have tried to apply some more modern methods, and I'm going to talk about those methods in a minute after I just share with you a little bit of what we found to explore whether using bedaquiline for longer than 6 months improved treatment outcomes. And this was an important question because, as I mentioned, bedaquiline's indication was for 6 months. It had been studied for 6 months in the pivotal trials there was no real reason for that. There had been no prior work to see whether 4 months was better or worse than 6, or 8 months was better or worse than 6. So these questions remained open even after the after the regulatory authorization. So in a paper just very recently published by Letizia Trevisi of the department and Molly Franke and our team, we did not find any evidence that prolonging bedaquiline beyond 6 months improved treatment outcomes within a regimen that was already effective. The analyses shown here are analyses that were inverse probability weighted and baseline adjusted in order to try to deal with many of the sources of bias that come into observational studies when there may be different confounding by indication. So the thicker people probably got longer treatment with bedaquiline and other sources of bias. And indeed, the different forms of analysis that Letizia did showed that there probably was some bias and that it was important to account for these sources of bias with appropriate methods in order to get reasonable estimates. Kind of analogous analysis that we did. This was done by Carly Rodriguez as part of her doctoral dissertation. She explored whether adding delaminid to an already effective 3-drug background improved treatment outcomes. And I'd mentioned that this was an open question after the phase 3 trial of delaminid had equivocal results. And we also did not find any evidence of benefit to adding delaminid when there was already a strong background regimen. We suspect that doing this analysis, kind of raising these sorts of questions of, say, extending clofazimine or adding clofazimine when you already had a strong background regimen, you might not see any benefit from all of these, and that what really matters is what's going on in the background initially. So I kind of gave you the punchline before. I'm giving you a little peek into some of the methods that we've been able to apply in the EndTB observational study to really raise the standard of what's done in observational studies for TB. We do have so many questions that don't get answered in clinical trials, and yet when we have good quality data, there's the potential to draw inference, and we don't always realize that potential. So using these methods allows us to ask questions like those I mentioned: which combinations are best or safest? What duration is best or safest? Are there specific characteristics that we can isolate that predict effectiveness and safety? And I'm just going to give you one example. It's the delaminid example. Does the use of delaminid improve treatment effectiveness? And it seems like a simple question, but it really has taken Molly Franke's leadership in applying these modern methods for us to address it in as unbiased of a way as possible. And I just want to express my gratitude to Molly, both for the work and for the slides that are going to follow. So this first slide is a heat map of 39 patient regimens, just randomly selected 39 patients from the EndTB observational study. And this axis gives you the number of days on treatment, and the color, the intensity of the blue reflects the number of drugs that the patient got at any time. So just by looking at this, you can see big changes over time. And you can imagine that the 6-month magical mark, even if you just look at before and after 6 months, there are differences. And then if you look at between 6 and 8 months, there are differences. And I'm just going to highlight this very bottom patient who at 6 months was receiving, I think it's 6 drugs, and by 8 months was receiving 3 or 4 drugs. So this is just a hint in when we say, did somebody get delaminid or not? That's a much more complicated question than we might be anticipating. You can see just this summary of the number of regimen changes in the EndTB observational study. So common in these longer regimens for MDR-TB. There were a median of 8 changes if you include just dose adjustments and temporary suspensions. If you exclude the dose adjustments, there was 6 median changes over the treatment period. And then if you think about the unique regimen combinations, there were still 5. So a tremendous amount of variability in these regimens. And the implication of that is that you really have to know what you mean by did somebody get delaminid, or asking the question, does adding delaminid help? And so a pretty common way to classify the exposure previously has been, did somebody ever get delaminid? And you can see that in all 6 of these cases, the answer to that question would be yes, but they all look really, really different. So this patient got it just for the first month. This patient got it for kind of a month in the middle. And that comes with some of the biases that I was mentioning. This another example in the way that it's traditionally been classified, that exposure has traditionally been classified as whether somebody got delaminid or the drug of interest in the first month. And you can see the answer is yes for all 3 of these patients, but no for this patient who in the end got 21 months of delaminid. So you do have a risk of misclassifying exposures in either over or underrepresenting exposure, depending on how the definition is used. And as I mentioned, there could be confounding by indication, where maybe this person started delaminid because they weren't having a good treatment response, or similarly here. And you need to know that. You need to know why it was stopped or started. It could have been stopped here because of toxicity or because of an excellent treatment response. And not having that information incorporated into the analysis can be the source of bias. We also just saw on kind of a population level changes in delaminid use over the life of the EndTB observational study. You can see that, oops, sorry, went too far. That if you just classified exposure based on whether or not it had been received on day 1 or it was ever received, you would have about a 20% difference in those classified as having been exposed. So how do you do the analyses, the fancy analyses that I showed you that Carly and Letizia did under Molly's guidance? So I think that probably the most important part of it is to clearly specify the question. And they're from our friends and causal inference next door at the School of Public Health, there is this target trial emulation framework, which really helps to specify the population and the exposure and the outcomes and what the comparison is that you're really asking about. And then they have also developed and adapted statistical methods that allow you to adjust for the confounding that occurs at baseline, which I think we're more familiar with, and confounding that can happen due to changes over time. Like I said, regimen changes that could be due to adverse events or to changes in somebody's clinical status. There is a very a generous approach to sharing the code and methods to do these kinds of analyses, including a free textbook recently published by the Causal Inference Group that can really help do these analyses. So moving toward the last part of my talk, I just want to share some of the bigger picture changes effected through the EndTB observational study, the contributions that we've made beyond the kind of narrow scientific contributions and methodological contributions. So among the 17 countries where the EndTB observational study was conducted, 16 of them now use bedaquiline and delaminid, some more routinely than others. The lone exception is the Democratic People's Republic of Korea, where unfortunately, since the COVID pandemic began, there hasn't been any treatment for drug-resistant TB, much less bedaquiline and or delaminid. And injectables are rarely used in the routine now. They are still occasionally used and used some for rescue regimens, but not as much in the routine as before the EndTB project started. We've seen some retention of the process of systematic safety recording and reporting, not just for bedaquiline and delaminid, but for TB treatment more broadly. I mentioned that the World Health Organization is kind of the arbiter of innovation to some degree for countries in their TB practices, and the EndTB observational study has contributed to 3 sets of World Health Organization guidelines, most recently last year, and represented a substantial portion of the data used for these guidelines. And the current recommended treatment composition for the longer regimen really reflects what we were doing in EndTB a few years before. The Global Fund and other bilateral and multilateral donors now support purchase of these newer and repurposed drugs because the WHO guidelines endorse them. And then the EndTB observational study platform has been a great foundation for the trials I mentioned, for the STEM observational study that Molly Franke is leading, and some other operational and implementation research that's being led by the World Health Organization, by MSF, both in EndTB countries and outside of EndTB countries. And then the last point here is just that we've been actively working to build the capacity through analysis of the EndTB observational study of local investigators on the project. Challenges. So despite all of these, all this great progress and the really substantial investment and accompaniment in innovation in TB care, we do have huge challenges. There is still just this, I don't want to say intrinsic because I have to believe it will change, but this hesitation to fully embrace innovation in TB. There are other examples of new innovations that haven't really been taken up. So preventive therapy, shortened preventive therapy for TB, its efficacy was demonstrated more than 10 years ago. It's very rarely used. The machinery around TB, which is very much a public sector machinery dependent largely on funding from outside entities, it does not move quickly. And part of it is this, what Jim Kim would call a failure of imagination, to think that we can do better. These WHO recommendations that I mentioned have been conditional. They have not been strong, and countries are slower to take up conditional recommendation. This whole effort, although now it's been a 9-year effort, I want to emphasize that the observational study was really just the first 4 years. And honestly, when you are trying to confront or overcome a legacy of colonialism and all of the disparities that come with that, a 4-year project is not enough to really accompany individuals and governments through a process of adopting innovation. I've mentioned the special populations. Their exploration of these special populations tends to be slower because they're in smaller numbers, and we really need more time and more data on those with comorbidities, pregnant people and children. The sharing of data to make sure it's usable to those who have produced the data in these high burden countries, it doesn't stop at the end of the 4-year period, and these kind of grant cycles don't support the longer term curation and sharing and supporting in training or analysis as needed. The last 2 points, just that the cost of innovation remains really high. I mentioned the cost of bedaquiline. It has come down to $340 in high burden, low income countries. But I just want to note that that is despite substantial public and philanthropic funding for bedaquiline's development and additional funding for like from USAID to buy up large quantities of bedaquiline and make it available for poor countries. A recent paper estimated that Johnson & Johnson philanthropic and public funding was about 1.5 to 5 times that of Johnson & Johnson in the development of bedaquiline, and yet they continue to use patent protection. They're filing for a patent extension as we speak till 2027, and restrictive licensing strategies that keep the costs of these drugs high. And I already mentioned a little bit the historical disparities and their ongoing consequences in inequality and the expectation that we can't do very much. So in summary, I think the EndTB project was a great example for us of service-linked research that can really be an important platform for improving care, for advancing policy, for improving research methods and doing this research accompaniment along the way. It does require proper long-term inputs that are going to be more than 4 years in a space like this. And just a summary of the scientific findings, we were able to demonstrate that both bedaquiline and delamanid containing regimens are safe and effective, that their concomitant use is also safe and effective, and that these findings held even considering the serious prevalence of comorbidities and extensive disease in the groups. And lastly, that it can be difficult to understand what little tweaks to the regimens do against a background of a strong regimen, but novel methods can do better than traditional methods with that. So I will stop there and stop sharing my screen, and I'm happy to take questions or discuss. Thanks so much.
There we go. Carol, thank you so much for that. And really just the value of evaluating these drugs in the real world and showing their utility in the real world, despite these considerable obstacles. And for anyone listening in, if you have questions, please submit them through the Q&A function. I see some coming in already. I guess one question I had, when we think of terms like accompaniment and research accompaniment, it also brings up these longer notions of accompaniment. And what in these observational trials, what was done to enhance quote unquote adherence to the regimens themselves?
So it was the kind of same model that we've used in the past, that it was providing nutritional support, providing social support, making it easier for people to get to appointments, providing transportation if that's what they needed, just basically being present with patients. Sometimes it meant bringing patients from a rural area to a location where these drugs were and finding temporary housing for them.
Thank you.
That range of supports.
Okay, so Lawrence Lucy asks, thank you, Carol and the team, for the great work you do for all DRTV patients around the world. Based on the findings of the EndTB observational study, do you consider a 3-drug regimen of bedaquiline, delamanid, and linezolid on the same level of effectiveness with the recently recommended bedaquiline TA, which you'll know what that is better than I would, linezolid for ex-DRTB patients?
Thanks, Scott, and thank you, Lawrence. So Lawrence's his photo was on my acknowledgement slide. Lawrence was one of the investigators in the EndTB observational study in Lesotho. So it's a pleasure to have you with us, Lawrence. And you could probably answer this question as well. So, many of you know I am not a clinician, and so I hesitate to make clinical recommendations, but based on my read of the evidence, I actually would not recommend a 3-drug regimen, either a bedaquiline pretomanid and linezolid regimen or a bedaquiline delamanid and linezolid regimen. We see a lot of toxicity, a lot of dose and duration limiting toxicity with linezolid, and the risk that basically means that people would be getting a 2-drug regimen, and that just does not seem to be a wise strategy given what we know about drug-resistant TB and about the bug itself. So I can't say anything about the equivalence of delamanid and pretomanid. For those who don't know, these are both drugs of the same class that were being developed pretty much contemporaneously, one by a Japanese manufacturer, delamanid, and the other by a private-public partner in the US. But they've almost never been compared head to head. They have only in like one tiny mouse model. There is a study coming up that will compare them head to head in a multi-drug regimen. So it will be interesting to see what the pan-TB study that Gates MRI is sponsoring will yield.
Thank you. An anonymous attendant, he asked two important questions. One, is there going to be any data that they might get on the development of resistance to the newer drugs?
Yeah, that is a really important question. So we do have limited data on this in the EndTB observational study. We tried to get repeat drug susceptibility testing and sequencing on isolates from people who either experienced relapse or experienced treatment failure. We are still trying to pull all of those data together. The sequencing was done by different labs. And then we'll have more information on that from the EndTB and EndTB clinical trials where we were able to capture, get isolates tested more systematically.
I should know that question was prefaced by, thank you for the great talk, and thanks so much for bringing great drugs to people who really need them through the study. Their second question, is there any data on outcomes stratified by nutritional status or BMI?
I think that I'm not sure. I'm going to answer honestly. I do not remember if we have done that on the whole cohort. I know that Lawrence is actually working on an analysis in Lesotho, but I don't think we've done that in the whole cohort yet.
And so last one gets at this question that you and I have spoken about with respect to endpoints and lung function, both of going into and coming out of a trial. But so this question by Dr. Kunda was, how is the effectiveness of the use of bedaquiline and lamanid shorter regimen on the extensive lung disease patient, if that can be operationalized?
Yeah, it's Dr. Kunda, that's an excellent question. Dr. Kunda is also an investigator working in Lesotho on the EndTB and EndTBQ trials. There is a growing body of evidence that the effect of shortened regimens in TB is modified by the extent of disease, and so that people who have more extensive disease, like on radiograph, who have higher bacillary burdens at baseline, that they don't do as well on shorter regimens as people who have less extensive disease. We don't really know the answer yet to that question for these all oral shorter regimens for drug-resistant TB, but it's definitely part of the plans for analyses in both the EndTB and EndTBQ studies.
Well, thank you. And certainly, I often lecture on the history of clinical trials, and it's incredibly complex now, in the real world, and you've done such an incredible job, you and your colleagues, in working your way through those complexities. So, Carol, thank you so much for a fantastic talk today and for all the work you and your team do. And for folks listening in, our next session will be on April 5th with Dr. Ananya Banerjee from McGill University, who will be speaking on the white savior complex in global health programs. So it should be a really interesting and provocative session. I certainly encourage you to join us. And Carol, thank you again for just a wonderful talk.
Thanks so much, Scott. Thanks, everyone. Have a good afternoon.